Hi, everybody. My name is Marley O'Brien, and I am tonight's host of the third annual Environmental Research Film Festival here at the University of Texas in Austin. Tonight, we have a lot of really awesome films to showcase covering environmental science students' independent research projects. The showcase will be broken down into three sections: films covering biological sciences, geographical sciences, and geological sciences. I now invite you to sit back, relax, enjoy the show, and hopefully learn something new. Our first section will be all the films covering the biological sciences track of environmental science.
Hi, I'm Madison, and for my short film, I'm going to talk about a project I did that's titled Evolutionary Relationships of Pet Trade and Wild Epipedobates. Epipedobates is a genus of poison frogs, and you can see here we have some nice pictures of the two poison frog species I studied. So first I want to give a little bit of background on these two species of poison frogs. So these two particular species, Epipedobates anthonii and Epipedobates darwin-wallacei, are sold in the pet trade. They're captive bred and raised. So the thing is that these relationships between species in the genus Epipedobates can be unclear a lot of the time, and our understanding is always changing about where they fall in relation to each other. Also, different populations of one species can look really different from each other, and populations that are different species can look really similar. So that makes it more difficult. Um, because of all this, it's possible that individual frogs sold in the pet trade are incorrectly labeled, especially Epipedobates darwin-wallacei. And actually, one wild population of E. darwin-wallacei was found to be almost genetically identical to the species Epipedobates espinosi. So to look at these species relationships, I first had to amplify this gene cytochrome B from the genomes of the two individuals. This gene is highly conserved in frogs throughout time, and I used PCR to amplify it. And there's a diagram about that right here on the right. So after that, I was able to sequence cytochrome B for the two individuals and align the genetic sequences, which then allowed me to construct an evolutionary tree and show how these two individuals are related to wild populations. And this on the right here is just a general phylogenetic tree for Epipedobates. So I found that the pet trade Epipedobates anthonii that I tested fell in with individuals from wild populations of E. anthonii like I expected. And most interesting was that the Epipedobates darwin-wallacei individual was placed with individuals from wild populations that are now considered to be a part of E. espinosi, as you can see on the left here. So that means that these frogs sold under the E. darwin-wallacei label should really be considered E. espinosi. Uh, so I'm going to talk a little bit about the illegal wildlife trade based on these results. So it was estimated that about $3.2 billion worth of wildlife is entering the United States illegally every year. And this is a big problem for species that are at risk of going extinct, like a lot of the poison frog species. So the way I see it, these methods I use for determining closely related populations could be used to determine where illegally imported wildlife is coming from, and then this could possibly lead to better enforcement of wildlife protections. So an example of this is if an illegal shipment of poison frogs is confiscated at a border or something, phylogenetic analysis can be done on the frogs and say they determine the frogs are the species Epipedobates anthonii, and they're most closely related to this population called the Pasaje population in Ecuador. So now, as a result, authorities can step in and monitor for poaching in this area. So the next steps based on these ideas would be to put pressure on national authorities to put resources into finding out the sources of poached and illegally imported wildlife. Then as a result, that would allow us to stop the illegal wildlife trade at the source instead of just relying on confiscation. And all that with the ultimate goal of protecting endangered wildlife and preserving the health of species at risk in our Earth as a whole. I hope this was educational and gave you a sort of different perspective on protecting our wildlife. Thanks for watching.
Hi, my name's Lauren, and this is my capstone project, Time to Eat. I looked at the latency to feed of Sheba's short-tailed fruit bats, Carolia perspicillata, at various reproductive states. So, the two main objectives was, how does the reproductive stage of a bat, whether reproductively active or not, influence the time taken to approach and eat the food source? And how does fruit ripeness impact the time taken to eat or the latency for these bats? So here, latency is the delay or time taken. So why do bats matter? Well, bats are the only mammals that can fly. They provide ecosystem services like pollinating plants, controlling insects, and aiding in seed dispersal. And Sheba's short-tailed bats are actually frugivores, which means they eat mainly fruit. So why are we doing this research? Well, first, in mammals, the reproductive and developmental state often influences foraging patterns and diet. Also, when bats are reproductively active, their energy expenditure during foraging and flying typically increases, meaning they spend more energy. Also, bat foraging habits are influenced by their nutritional and energetic needs. So, if a bat is reproductively active and is spending more energy, the food they acquire and how they acquire it may differ. Lastly, climate change impacts plant phylogeny and bat migration, meaning it could potentially alter the bat-plant interaction we see today. So, why is this research important? One, we get to gain better understanding of this relationship between these fruit-eating bats and their food source and complete further research on this relationship and the role they play as seed dispersers. Also, we gain more insight on the correlation between reproductive state, latency to feed, and fruit ripeness. Lastly, we're able to see any changes in how the bat-plant relationship occurs over time due to climate change. Methods. To test this, we housed 26 different fruit bats, all different reproductive states, and manipulated their food source. So each bat was individually housed in a cage over 3 different nights and recorded for 30 minutes. For this project, we got to observe these videos and collect data on their behavior and compile it into a master spreadsheet. And then here is a clip of the fruit bat flying. So what we expect to see. Our first hypothesis is reproductively active bats will have shorter feeding times or latencies than non-reproductive individuals. Also, all bats, regardless of reproductive state, will approach unripe and ripe fruits faster than overripe. The current status of this project is not yet complete. We are still scoring videos, and I have yet to complete a large enough sample size to draw any significant conclusions. But data analysis will be complete by fall 2023. Thank you, and I hope you enjoyed.
Hello, my name is Michael Adam Jones, and I'm going to talk a little bit about the colony size regulation of American cliff swallows, one of which is shown here as it glides along looking for insects to eat. So just for some background on this species, cliff swallows are very common and widespread in North America, often forming large colonies on the culverts of highways, like the one shown here, on the right. What makes the colonies of this species so interesting is that the use of colonies is not very common among terrestrial birds. Colonial behavior generally occurs more in marine bird species. Because of this uniqueness, understanding these colonies could also help us better understand how coloniality develops. Additionally, understanding this important aspect of cliff swallow behavior could have management implications, not only for cliff swallows themselves, but also for other species they come in contact with. One of the most significant possible impacts on other species of cliff swallows is their larger potential for the development of diseases, as their colonies often support extreme density, which increases the spread and evolution of pathogens. So with this background information in mind, the goal of this study is to investigate the environmental factors that could affect changes in cliff swallow colony size. These include the relative populations of parasites within colonies and several factors relating to the access of resources around the colony. This assessment would ultimately give us insight into what determines the habitat suitability of a given colony for cliff swallows. Further analysis of the effects of these factors would also have to address the mechanism behind them, whether that be through the change in cliff swallow behavior or a change in their relative success or mortality. We expect that habitat suitability, as assessed by these environmental factors, would result in a related non-random change in colony size. And for this investigation, we are gathering non-invasive samples of parasites from select nests, which is what I'm doing on the left images here. These samples, along with measurements of other environmental factors, will be used to compare within colony variation in the change of the number of nests, as well as variation between the two colonies of interest, which are both shown here on the right. This analysis will ultimately allow us to assess which factors are likely to be the most significant contributors to this variation, and allow for further investigation into those factors. So I just wanted to thank you all for your interest in this project, and I hope you learned a little something about a commonly seen bird in North America. Bye-bye.
Hello everyone, my name is Alejandro. I'm an EBS Bio and my project is Birds in Our Noisy Cities. Okay, so a little bit of background. An urban ecosystem is a new ecosystem that is mostly dominated by our buildings and activities, but have communities of plants and animals living within. These are our cities, residential areas, and so on. Picture here is one example of an urban ecosystem. It is Central Park in New York City. So within these cities, we release noise pollution that causes annoyance and irritation to both us and other animals. This noise mostly comes from our cars, airports, and even landscaping, like our leaf blowers. So this starts to present issues, as birds rely on acoustic vocals to communicate with each other and use certain songs to find a mate to reproduce. As you see in a picture, there are still plenty of birds around, but they always seem to be the same one, the grackles. This led me to my project question. What are the effects of noise pollution on bird diversity? So in order to test this, I will be collecting data this summer by using point counts, where essentially I will stand in one point and telling how many birds and how many types of birds I spot within 20 minutes. This will repeat it about 2 or 3 times. With these point counts, I can see how many different species are present at a site and their relative abundances. This will be done at 10 different sites within the green spaces in Austin. Those sites will capture a spectrum of noise from downtown Circuit Park to a more residential Walnut Creek Park. Here's an example of what a site will look like. This was taken at Circuit Park near the hiking trail. So noise pollution, along with other confounding variables that can influence bird diversity, will be measured. And so my hypothesis is that an increase in noise pollution will lead to a decrease in bird diversity. So once I've gathered all the data, diversity at each site will be evaluated using the Shannon Diversity Index that is essentially able to quantify diversity based on a number of different species, which we call richness, and the relative abundances, which we call their evenness. So I will use R to run a multiple regression model that will test each confounding variable, including noise pollution, to see if there's any significant correlation with bird diversity. So why is this important? Well, different birds play different roles, from pollination from hummingbirds to controlling population of insects from songbirds. More than that, they're just beautiful creatures of different colors and shapes, with some singing elaborate songs that we all can enjoy. Here are just some of the cool birds we see here in Austin. So the impacts of noise pollution on different birds has not gotten much attention until recent years, but scientists have already found that it can lead to behavioral changes in birds, like singing at earlier times. For this reason, and the potential it has to affect bird diversity, we must come up with creative solutions to combat our noisy cities. Thank you.
Hello, my name is Marcus Fundora, and my upcoming capstone research project is entitled Austin Area Water Phosphorus Levels. To get started, I would like to introduce some necessary background information about why this topic is worth discussion. Water phosphorus levels are the primary cause of eutrophication, which is what happens when phosphorus levels are very high and the algae in the water increases way above its normal population levels. This bloom of algae then requires an increased amount of oxygen in order to sustain itself, depleting the oxygen for the rest of the species inhabiting the water source, devastating them. The usual causes of phosphorus level increases are human agriculture and human waste or pollution. Austin as a community has grown rapidly over the past 2 decades, as the human population of the city has more than doubled from 1 million to 2.2 million people. This project will be based on a previous study, Austin Area Phosphorus Levels, by Martha Turner. She took samples of water phosphorus from creeks, springs, stormwater, rainwater, and other sources to compare the phosphorus concentrations of each. She also compiled previous data about Austin phosphorus in order to see how much it's changed since the 60s. Some of her results include finding high dissolved phosphorus levels in urban stormwater, particularly around single-family residential areas and golf courses. She has also found that the average level of phosphorus in creeks was higher than needed for eutrophication to occur. Her final major conclusion was that phosphorus levels had been increasing in most areas studied, with stormwater being the only exception. With all of that in mind, my research question is, how have Austin's water phosphorus levels changed over the past 20 years? My hypothesis is that due to increases in human activity over the past 20 years, water phosphorus concentrations will be higher than they were previously found in Turner's study. The methods which I will be testing this hypothesis are as follows. First, I will be conducting my own tests of various sites of creeks, springs, rain, and stormwater with handheld water samplers. Each site will be categorized by certain properties, like how urbanized the site is, or by how long it has been since the last rainfall. I will also be looking at organizations that collect phosphorus data in the area as well, and comparing their trends to the ones observed in Turner's study. Thank you for your time, and I want to give a special thanks to Daniel Levine here at the University of Texas for helping me through the steps necessary to get this project going, and thank you to the amazing people who helped put this film festival on.
Today, most Americans spend a lot of time driving. But we need a lot of tires to do all this driving. And they are prone to wearing out. With many of our cities and towns near the coast, or near river systems that lead to the coast, a lot of harmful toxins enter our waterways via runoff. A lot of this runoff comes from impervious surfaces like parking lots and roadways, causing the introduction of pollutants to our rivers and oceans. One of these pollutants is 6-ppdq, which comes from chemicals found in tire wear particles, and has been cited as very highly toxic. 6-ppdq has been linked to incidents of urban mortality syndrome in salmon. So there is concern that this pollutant threatens other fish species. One species of concern for Texans is the red drum, because it is very valuable for sport fishing in the economies of coastal communities. Red drum are dependent on estuaries for feeding in nurseries, and are more likely to encounter contaminants from runoff sourced from Texas rivers. My study focused on testing red drum larvae sensitivity to 6-ppdq, measuring body parts after exposure to various concentrations. Larval success is highly dependent on good eyesight, proper brain function, and good body proportions. For marine fish, being bigger means having a better chance at survival. When these fish are in a stressful environment coping with toxins in the water, more energy will be spent on repairing damaged tissue and being ill-equipped at avoiding predators, as opposed to normal tissue development. If they are very sensitive, generations of unsuccessful larvae could spell disaster for red drum and lead to population collapse. During this research, I found that they have an LC50 of about 200 ppb. This is the concentration at which 50% will be dead. To put this number into perspective, mass casualty events of coho salmon occurred at about 1 ppb, so red drum are not very sensitive to this pollutant, which is very good news. Now that we know their sensitivity, we need to develop methods for measuring this toxin in our estuaries and bays to determine real-world risk. We should also expand sensitivity studies to other Gulf species and determine their risk. With this better understanding of 6-ppdq, should we change how we manufacture tires and other road products? Should we monitor biomagnification in our seafood? 6-ppdq is very new to marine toxicology and requires continued research to address these questions.
Hey everyone, I'm Joji Sherman, and like many others tonight, I'm a fourth-year student here at UT. Over 2022, I focused on exploring new ways to detect sewage in urban waterways. In an aging city like Austin, many of our sewage systems are beginning to fall apart, resulting in leaks. These leaks dump pollutants and disease directly into the waterways sewers are built around. Our drinking water risks contamination. Areas where Austin wants to establish recreational zones and cultural centers for us to enjoy remain unsafe. In short, it is a public health issue that prevents us from enjoying Austin to the fullest, or living there to the fullest. Of course, ideally we could repair the entire infrastructure, but that is more than a bit expensive. In the meantime, we need to settle for repairing individual leaks, but we have to detect those first. That's where this project comes in. Sewage detection has been investigated for years, but every method has some problems. Oftentimes people have introduced dyes into sewerage systems suspected to be leaking before tracking their presence in the nearby waterway. Bulk quantities will offer color to be directly observed in the water, but come with pollution issues themselves, while small quantities can be detected through a detection method known as fluorimetry. For a basic summary of fluorimetry, you put a specific color into a sample, and light of a different color comes out depending on the amount of chemical present. With the right machines, even small amounts of this released light produce a signal, as seen by the peak here, meaning extremely low concentrations of dye can be detected. However, there are also issues that will be explained in a bit. For now, let us return to Austin, and in particular Water Creek, the waterway that runs through the UT campus. Based on previous tests done by the UT Environmental Health and Safety, we released a red dye, eosine, and a yellow-green dye, pyridine, into 3 buildings suspected to be leaking, labeled on this map as MRH, LBJ, and DFA. We then collected creek samples from the suspected leakage outlet in addition to upstream control sites to determine if there was a leak and later which building the leak came from. And hey, leaks were detected as seen in this graph here. However, similar signals were obtained at the upstream site and control samples, suggesting one of two things. There is another leakage outlet or other chemicals in Water Creek are interfering. To determine if there are interfering chemicals, that is where the next step, fluorimetry kinetics, comes in. While in some instances, multiple chemicals release light of similar colors, they may release it at different times. And we can look at the time delay between the release in order to tell which chemical it is. While such methods were not directly implemented due to time constraints, the project so far has showed the usefulness of doing so. Where do you all come into this? Well, to be frank, a lot of this is technical research, so it may be hard to directly involve yourself. However, educating yourself about Austin research and creek initiatives will equip you with the knowledge and resources to aid in bettering our city's environment and public health.
Nature. There's just something about the sound of birds chirping and flowing water. But in the middle of a city, urban ecosystems like Waller Creek here provide valuable ecosystem services. One of those should be flood protection. But since we designed cities to channel all water directly to creeks through impervious surfaces and storm drains, an urban creek can quickly become a hazard during even small rain events. Austin creeks naturally flood pretty easily, but urbanization makes their floods quicker and more intense. This has led to the city of Austin to undertake a massive restoration project on Waller Creek, a flood diversion tunnel to divert stormwater underground and into the lake, which has dramatically decreased the floodplain downtown.
And that's where my research comes in. Hi, I'm Ashley, and I've been studying how primary production is altered by both urbanization and this flood diversion tunnel.
Algae are important because they are the base of a creek's food chain. As primary producers, they turn carbon dioxide into oxygen that other organisms like fish can use. When it floods, algae can quickly be wiped out in what we would call a scour event, and then they have to regrow all over again. I'm interested in how the scour and then recovery might be different between urban, rural, and managed creek flows.
I measured the amount of algae before and after rain events to see how the primary producers are impacted and then recover from these stronger and faster storm flows.
To do this, I collect algae samples from 3 different creek ecosystems: my urban sites at Waller Creek upstream the diversion tunnel, my managed urban sites on Waller Creek that are downstream of the diversion tunnel, and finally at Bear Creek, a much less developed creek in southwest Austin, as my rural site. One of the ways that I process these samples is by extracting chlorophyll A, a molecule found in photosynthesizing organisms like algae. Then I'm able to compare results between sites for a rain event. We can see that the algae in the urban sites regrow much quicker than the other sites. We can also see that the managed sites have pretty low amounts of algae both before and after the rain event. That's a trend that I've seen in a lot of my data, suggesting that the higher base flow and maybe even sedimentation restrict algal growth, and therefore is not currently conducive to ecological restoration. Studying the impacts of this large-scale restoration project is important for the future of Waller Creek's ecosystem, but our results can also help to inform urban creek restoration across Austin and around the world.
Stingle Lost Pines Biological Station is owned by the University of Texas. A forest fire occurred at Stingle Lost Pines in August 2022. This station has several sites of water that have never been tested, including a stream and 3 ponds. This fire impacted the trees at varying amounts of severity around these bodies of water. As a result, we can measure the rate of recovery of the trees and how it impacts the water quality in the surrounding areas. I collected a water sample before the fire occurred and then collected samples periodically after the fire to analyze the changes to the water quality as a result of the fire. I looked at pH, conductivity, total dissolved solids, nitrate levels, and phosphate levels at 6 sites. I collected samples 7 times total. After data analysis, I found that the pH was consistently more basic after the fire. I also found that the conductivity and total dissolved solids both tended to increase after the fire. Lastly, I found that the nitrate and phosphate levels varied too much to be analyzed, so further analysis will need to be done to get a better understanding of nutrient levels at Stengel Lost Pines. Conductivity and total dissolved solids are important water quality parameters because they indicate the level of salts that are in the water. Salts are normal in water, but higher levels indicate contamination or dangerous conditions. Additionally, the pH is an important water quality parameter because some organisms can only survive in certain pH ranges, so understanding how pH is changing and how quickly can also indicate contamination and unsafe conditions for ecosystems and organisms alike. Lastly, nitrate and phosphate levels are important to understand and analyze because high nutrient levels can lead to algal blooms that deplete oxygen levels. High nutrient levels are also typically a result of contaminated runoff, so it is important to understand these parameters. Overall, the fire forever altered the forest structure, forest succession, and water quality at Stengel Lost Pines. I hope that this study encourages people to care more about water quality and the impact of forest fires on water quality and the environment. Understanding how natural disaster events impact water quality and other environmental issues is extremely important to understand how the world works and how we can better the damages humans have caused and are continuing to cause. Thank you.
O little forams, what do you hold? What are the secrets of the times of old? Foraminifera are single-celled creatures that have lived in the oceans for hundreds of millions of years. These crafty fellows are able to build shells out of their marine habitat. For instance, some foraminifera build their shells out of sand. Others build their shells out of calcium carbonate. When they die, the foraminifera's shell will sink to the ocean floor and fossilize. Scientists are interested in finding these fossils to learn more about the past. What the shells are made out of can tell researchers a lot about how these creatures responded to ancient global changes. Higher diversity indicates more favorable conditions for the foraminifera, while lower diversity indicates less favorable conditions. When plotting the diversity of foraminifera living at the bottom of the ocean, one finds interesting trends throughout the geologic record. For example, foraminifera diversity doubled across 36 million years, a mere blink of an eye in geologic terms, at the Devonian and Carboniferous boundary. The early Carboniferous was a period when atmospheric carbon decreased and there was a higher diversity of marine life. In contrast, foraminifera diversity more than halved at the end Permian mass extinction, circa 251 million years ago. This mass extinction is characterized by a high emission of carbon dioxide into the atmosphere, and 90% or more of marine life went extinct. In the Cenozoic, the era that we are in today, foraminiferal diversity begins to stabilize. While there are cycles of increased and decreased diversity over the last 66 million years, the changes are at an expected rate. Today, foraminifera diversity has over 800 groups of species. The past provides prologue for present and future events, and understanding the global trends of foraminifera through time provides crucial information for other organisms like corals and shellfish, which are also vulnerable to changes in their marine environment. By contextualizing ecosystem responses to atmospheric carbon throughout the geologic record, marine conservationists can project future responses to atmospheric changes. Thank you for watching, and remember, nothing is too small to make a big impact.
Climate change. It's happening, and it's happening fast. So fast that plants all over the world are becoming incredibly overwhelmed with all the added pressure. These changes are demanding plants to acclimate quickly enough to survive, but many can't keep up with the intensifying pressures. So we're seeing a shift in plant community composition, distribution, and abundance. All of this added together is increasing biodiversity loss and altering ecosystem dynamics. In order to tackle this issue, we need to first understand how exactly a plant acclimates to its new environment. One way is to study trait plasticity. This is the ability of plants to alter their physical traits, like leaf or root production, in response to external stimuli, like droughts, to cope with their changing environments. Because plant responses are super complex, trait-based approaches enable us to quantify these mechanisms, giving us predictive power in understanding how exactly plant communities will change. But these studies tend to focus on adult plants that already have the mechanics to survive. So what about the little guys? The ones who are still just trying to figure it all out? That's where I come in. My name is Casey Din, and this year I wanted to turn my attention to seedling trait plasticity because it's a topic that hasn't been widely explored yet. I'm joining forces with the ongoing plant biodiversity experiment conducted by Dr. Wolf and her graduate students. This is held at the experimental gardens of the Brackenridge Field Laboratory. The seedling stage is the make-or-break moment, when a plant is most vulnerable to mortality. Seedlings need to prove themselves against factors like competition or overcrowding. In a seedling population, this is called the bottleneck effect, wherein only a handful of them actually make the cut. As for the rest, it isn't a very pleasant tale. With all this in mind, my study aims to investigate what factors significantly influence seedling mortality. For my study, I've selected 3 Texas native species: the Texas plume, lemon bee balm, and blue curls. To set this experiment up, I tagged seedling individuals either in plots of 4, 6, or 12 species. In other words, I've manipulated the species richness levels. For each species, I tagged 15 individuals per plot, 5 of which were tagged in a different color to be harvested later. For all individuals, I observed mortality and growth rates over 2 months. The 5 individuals that were harvested, I measured these 6 seedling traits. Now remember, we wanted to investigate the relationships between seedling traits and mortality, or species richness and mortality. Therefore, I'll be running multiple linear regression models to see if there are significant differences among each trait in mortality or growth rate, and among species richness and mortality. I expect that there will be significant trait differences between low and high species richness groups, along with higher mortality rates in plots with lower richness. These expectations stem from the idea that if individuals in a small area share the same niche, they overlap. As a result, they'd be brawling it out to compete for the same resources. However, if you diversify the plot, they'll have a little more wiggle room to compromise. So why does all this trait plasticky stuff matter anyways? Well, frankly, it's because you matter. Plants are the foundation of life itself, so maximizing our knowledge on seedling trait plasticity will open more doors to understanding how our plant communities will change in the future. So with all that being said, may the best seedling win.
Hello, my name is Bryn Havern, and I'm currently a junior environmental science major with a focus in biology at the University of Texas at Austin. Though this film festival is meant to highlight students' capstone projects, I will not be starting my capstone research until next semester. Instead, I will be highlighting a topic I'm very passionate about, which is the impact of native bees on American agriculture. First, I will provide a little overview of bees in the United States. The pollination effort of thousands of species of bees across America are responsible for about a third of the food we have on our plates. European honeybees are the bees most people are familiar with. These are the bees that produce the honey we consume and that live in hives that beekeepers maintain. Despite these bees being important pollinators for American agriculture and the economy, they are non-native and considered invasive. Many American farms use honeybee hives to pollinate their farm, many of which are large scale and have no diversity in crops. This is called monoculture. Many managed honeybee colonies that are used for large-scale American agriculture have been lost due to colony collapse disorder. Colony collapse disorder is directly linked to a lack of nutrients that bees receive from monoculture. As many farms lose their European honeybee hives from CCD, some farmers are turning to native bees for pollination services. There are over 4,000 species of native bees throughout the United States. According to the USGS, between 20 to 45% of all native bee species are pollen specialists. You may be asking yourself, what is a pollen specialist? Well, a pollen specialist is a pollinator that only pollinates a certain type of flower. For example, the squash bee is a U.S. native bee that only pollinates the flowers of squashes. These bees have circadian rhythms that wake them up in order to pollinate the flowers of squash very early in the morning, the only time the flowers open, and a time when European honeybees are still in their hives. Squashes, pumpkins, gourds, and sunflowers rely heavily on native bees for their pollination services. The carpenter bee is one of the most productive pollinators of tomato plants. These bees are all examples of why native bees can play such a crucial role in American agriculture and food diversity. In 2014, a study examined the effects of diverse wild bees on apple orchards compared to introduced European honeybees. The study examined factors of bee abundance, bee species richness, and apple fruit set. Fruit set is when ovaries develop into fruits after pollinators have fertilized the plant. Half of the orchards observed had managed honeybee hives, and the other half did not. The study found that honeybees did not create a higher fruit set. Instead, there was a large fruit set increase due to diverse wild bee populations. This is related to the fact that honeybees tend to only pollinate apple flowers that occur in bunches, whereas wild bees will pollinate apple flowers equally, no matter the spatial patterns. This graph shows the relationship between wild bee species richness and the proportion of fruit set. The study displays that native bees can play a crucial role in increasing crop yield for American farms and are often more productive pollinators than European honeybees. Thank you for taking the time to view my film and learn about the research I am passionate about.
What can I do about climate change? As an aspiring environmental scientist and ordinary earth enjoyer, this is the question that's always on my mind.
This year, I'm channeling my climate anxiety into researching how plants defend themselves against their natural enemies so we can better protect them as environmental conditions change. Okay, so get this. Pollination is not the only interesting plant-insect interaction. Plants also have antagonistic relationships with insect herbivores, which means that part of their survival strategy includes defending themselves against getting eaten. However, plants can't just spend all their energy on defending themselves. They have other things to do.
Plants gain energy in the form of carbon through photosynthesis. But after photosynthesizing, they are faced with a critical decision. How are they going to invest the limited amount of carbon that they have? Well, when faced with insect herbivory, I hypothesize that plants will allocate some of their carbon to defending themselves at the expense of using it to grow.
The problem is, we don't really know how plants decide what to spend their limited amount of resources on. But what we do know is that plants and insects don't exist in a vacuum. In real life, they're also interacting with the organisms around them. So, what if plants make different decisions based on who their neighbors are? That's where the experimental gardens come in.
Here in the gardens, I am investigating whether plants will experience different rates of insect herbivory depending on the diversity of their plant neighbors, and whether they will choose to invest more resource into growth or defense as a response. To do this, I am going to tag individual plants living in plots of low, medium, and high plant diversity. Then, I'm going to measure the leaf areas and herbivory defense traits of my tagged plants over time to see whether they will invest more into defense and less into growth within the plots that they are more heavily munched on by insects.
Plants are the foundation of our ecosystem. As more vegetation is lost due to climate change-related stressors, the existence of insects and of all the other organisms that depend on plants is also threatened. This is why understanding the mechanisms behind how plants interact with their changing environments is essential for us to protect our wildlife.
By saving our plants, we save everything that depends on them too. And we start this by studying them.
Science is often a collaborative effort, and large-scale experiments conducted across global grasslands has created a network of ecological data for scientists around the world to use, known as the Nutrient Network. There is also Dragnet, a study that applies physical disturbance and nutrient addition to plots to investigate how these factors impact plant community dynamics and also aid in predicting how disturbance may impact grassland ecosystems in the future. My project builds off of the Dragnet site established at the Lady Bird Johnson Wildflower Center in Austin, Texas, where I am investigating the impact of nutrient enrichment and other climatic variables on insect herbivory of two grassland plants, little burr clover and the deer pea vetch. Herbivory is the consumption of plant material by animals, and nutrient addition into an environment can come in a variety of forms, such as applying fertilizers on lawns or agricultural activities. Prior research has shown that nutrient addition increases herbivory and decreases plant diversity within environments. Nutrient enrichment also impacts insect performance by decreasing the number of species but increasing the number of insects per environment. Prior research has also shown that in the long term, climate factors like precipitation and soil moisture can impact herbivore performance if conditions are too dry or too moist. Herbivory can also severely inhibit growth because plants will use their energy to build up defenses rather than growing, which can have negative impacts on other plant functions. Research for this project took place over a 5-week period where a total of 40 individuals were observed across 10 plots. 5 of the plots were treated with nutrient enrichment, composed of nitrogen, phosphorus, potassium, and micronutrients. The other 5 acted as controls. Preliminary results showed that there is a statistically significant correlation between treatment type and percent herbivory, and that individuals treated with nutrient enrichment appeared to show greater amounts of herbivory over time. Further analysis needs to be done. However, I hypothesize that nutrient enrichment also increases growth, and factors like soil moisture influence the amount of herbivory. It is important to study these plant-herbivore interactions because it can provide key insights into how insects will interact with plants in a climate-stressed future. Increased uses of fertilizers and other chemicals may improve plant growth in the short term, but in the long term can reduce biodiversity and degrade the landscape beyond recovery. Efforts to reduce pollutants like nitrogen in ecosystems are important to preserve plant-herbivore interactions and prevent a global decline in grassland plants.
Polyethylene terephthalate, or PET, is the most abundant polyester manufactured in the world and has been widely used for beverage bottles, packaging, clothing, and carpeting. PBS International makes pollination bags made of this PET material. They sent in samples to the Alpert lab at UT Austin, which created the successful fast-pedase enzyme that quickly breaks down plastic into smaller parts in a process called depolymerization. This bag made of PET is created by randomly laying down polyester fibers and then bonding them together without the use of chemicals. This material is referred to as Duraweb and is favorable to prevent cross-contamination during plant breeding. Duraweb also maintains its shape when wet. The overall purpose of this pollination bag is to help gardeners control weeds and erosion while providing breathable and durable protection. For my capstone project, I aim to evaluate enzymatic plastic recycling capabilities using the fast-pedase and LLC enzymes to degrade the pollination bag. I started off by hole punching pieces of the pollination bag into smaller pieces that would fit into the test tube. In order to evaluate the enzyme, the first trial took place using the fast-pedase and LLC enzymes with and without surfactants. Here are the results of the monomer concentration of TPA and MHET after a 3-day trial. In order to demonstrate a fully circular economy, repolymerization was attempted. Through adjusting pH levels, you can see the monomer being disassociated by adding a solution of sodium hydroxide. A final trial was completed with the highest yield of monomers, which was a heat-treated fast-pedase mix with surfactants. Further research should be done to fully evaluate the capabilities of these enzymes in order to decrease pervasive plastic waste that can last in our environment for over 400 years.
Inadequate air pollution mitigation in Texas. Picture yourself walking throughout your neighborhood park. As you take in a deep breath, you feel the sun kiss your face, the wind gently tousles your hair, and you revel in how wonderful and revitalizing the air feels. For some Texas citizens, they do not get to experience this clean air. Texas's inadequate air pollution mitigation has created hotspots in heavily industrialized cities and their surrounding areas, which poses a threat to the well-being of Texas citizens and their environment. Large emissions can create hotspots, which are areas that contain higher concentrations of hazardous air toxins. Exposure to these toxins can cause adverse health effects, such as respiratory illness, heart disease, cancer, and death. In 2015, Governor Greg Abbott restricted local governments from being able to enforce pollution laws and put a cap on penalties for pollution lawsuits. Also, between 2008 and 2018, funding was cut for state pollution control programs by 35%. TCEQ and EPA need to prove that a particular hotspot is the result from excessive emissions from one or multiple facilities in order to penalize them. This is difficult to accomplish when these agencies receive minimal funding and have their enforcement powers stripped away by the Texas government. EPA states that it strives to protect people from excess cancer worse than 1 in a million. However, EPA policy has set the upper limit for permissible excess cancer risk at 1 in 10,000, which is 100 times more than EPA's aspirational goal. In 2015, a Shell chemical plant spewed over 250 times the legal limit of carcinogenic compounds into the atmosphere. In response to this, the TCEQ fined them only $25,000, which is the maximum fine allowed. From 2012 to 2016, the Keystone Gas Plant had over 900 excessive emission events, and they were only fined once by the TCEQ. These facilities are not attempting to mitigate their emissions because it would cost them more to shut down a plant and perform upgrades than to pay these fines that are handed out. Of all the excessive emissions reported from 2011 to 2016, only 2.4% of illegal air pollution releases were penalized. The total fine for these violations was $13.5 million, which averages out to 3 cents per pound of pollution. TCEQ and EPA have a lack of funding. If these agencies had larger budgets, they could build more air monitoring systems, employ more enforcers, and hand down greater pollution penalties. All citizens of Texas deserve clean air, and it is the government's responsibility to protect Texas citizens from harmful air pollution.
I hope y'all are all having such a great time watching these films. I sure know I am. We're going to take a brief intermission for a few minutes before we start the next section. Hey y'all, weren't those projects really great? I'm excited to learn more about the geological sciences in this next part coming up.
You and an ant are no different to the boulders of the Guadalupe Mountains as you climb through its great channels. These boulders have been there for thousands of years, and you are both just passing through. Trying to understand their history and what they might look like thousands of years into the future is a noble pursuit, but it's one that takes patience and an understanding of how these boulders shape the land. Many previous geologic journeys have shown that the shape of channels through the topography is altered to accommodate the presence of boulders. They've shown that boulders control river erosion rates while causing the flow to veer off its original course. And yet, there's more to be learned. Isolating any given parameter in this complicated scenario remains difficult and is not well understood. Here we will take our own short geologic journey together to explore the influences that boulders can have on channel steepness in the Guadalupe Mountains of New Mexico and West Texas. In doing so, we hope to improve others' ability to understand the relationship between rock characteristics, or lithology, and topography, or the shape of the land. As we travel through the arid Guadalupe Mountains, imagine this place as it was 260 million years ago, a Permian reef. That reef has since been lithified and left us with a gorgeous deposit of carbonate rocks in an area that is tectonically stable and lithologically uniform, creating the prime location for our exploration. Although these channels alone hold so many secrets, Google Earth imagery, ArcGIS, USGS national map data, and MATLAB allow us to more fully understand the interactions in the landscape. By collecting boulder diameters and concentration, as well as steepness along 5 channels in the range, we can better understand just how much of an effect boulders have on the topography that we, and our ant friends, experience as we hike through the mountains. Although we see scatter in this relationship, 4 of the 5 channels show some positive correlation between boulder concentration and steepness. But the story isn't over. Channel evolution is complex, and we cannot say definitively that boulders are the sole control at play. There's always more to explore and always more to learn. For our next journey, we may live vicariously through a drone or use higher resolution satellite imagery while making use of data collected in the field. All in the hopes that we may get closer to defining a significant positive correlation between boulders and channel steepness while improving our understanding of the history of these landscapes to better understand the future shape of the land.
Do you not like sand? Is it coarse and rough and gets everywhere? Neither does life, which is why deserts are so hard for life to survive in. Yet for millions of people in the southwestern United States, the hostile conditions of the desert are a facet of daily life, and this number may be growing along with the desert itself. As such, it's important to understand how the desert is changing over time in order to know where it's heading, and how fast in order to prevent large amounts of economic and biological suffering. Even if it may not impact everyone directly, desertification will have a wide array of effects, from changing rainfall patterns to more expensive bread at your local market. But how do we tell if an area is desertifying? For such a large area, it's impossible to take measurements manually while accurately representing the region. The solution is remarkably simple: remote sensing. Using remote sensing, one can analyze a massive amount of data over a large area, as well as over long time spans. This is perfect for analyzing desertification as the desert moves over the course of decades to centuries, so any analysis of their spread must also be performed over the course of a few decades at a minimum. For this study, approximately 23 years of remote sensing data was analyzed over the American Southwest to determine if the desert was spreading. In particular, the region of study started at the western border of New Mexico and stopped at the 100th meridian, a boundary line within the United States where crops on its western side can't grow without large amounts of human help. The northern boundary of this study was located around the top of the Texas Panhandle, and the southern end being located approximately 30 kilometers north of the town Laredo in Texas. Desertification was measured using photosynthetic activity in the study area. By using photosynthesis as a proxy for the amount of vegetation in the study area, we can state with a degree of certainty that if photosynthetic activity decreases over time, it means that the amount of vegetation has decreased. As vegetation cover decreases, the warm temperatures in the area allow the desert to spread eastward into Texas, desertifying the region. After analyzing the data, the results are in. Photosynthetic activity in the region has been steadily decreasing over the study time span, which implies that it is desertifying. However, further work needs to be done to understand how intensely and quickly this desertification is occurring. But we don't have time to just wait around. This desertification will cause more intense desert storms, potential species extinction, and harder times growing crops. It will not change things for the better. Yet even if this desertification cannot be stopped, we have an obligation to slow its spread here and now, for those that live in these deserts, and those it looks poised to consume.
Hi, I'm Haley. I am a fourth-year environmental science, geology, and geophysics major. Over the past few years, I've been working on a project to try to see if there is a relationship between the 11-year solar cycle and El Niño Southern Oscillation, or ENSO. A little bit of background, ENSO is a natural phenomenon that happens in the Pacific during wintertime, and the Pacific Ocean has these three states. So there's neutral, warm, and cold. These warm and cold states are what El Niño and La Niña are. They occur every 2 to 7 years, and they have the ability to impact climate worldwide. We'll start talking a bit about El Niño. This is the warm state of ENSO. What happens here is that the winds in the tropical Pacific Ocean will slow down. This causes a buildup of warm water in the east near California and South America, and cold water to build up in the west near Australia. This causes flooding in California and drought and wildfires in Australia. On the other hand, we have La Niña events. These are coolest state of the Pacific. It's essentially the opposite of what happens during an El Niño event. We'll see drought and fire in California and a lot more flooding in Australia. El Niño and or La Niña events will occur every 2 to 7 years, so I was very interested to see if it had a relationship with the 11-year solar cycle that you see here. Over the course of the solar cycle, we see that the sun goes through periods of high and low solar activity. During periods of high solar activity, more energy is being released from the sun, and that energy is making its way to Earth. During periods of low solar activity, the Earth is getting less energy from the sun because it's producing less energy. There are two possible ways that the solar cycle could impact El Niño. The first is the top-down mechanism. This begins with the solar signal up at the top of the stratosphere, and through a bunch of atmospheric feedbacks, it works its way down. The bottom-up mechanism is direct heating or cooling of the ocean in response to the solar cycle. Both of these are thought to change atmospheric circulation, and it's very likely that they're working together. Let's take a look at what happens during the course of an 11-year solar cycle. So starting at the very bottom, where there's a solar minimum, we see this kind of cold La Niña-like relationship occurring, which you can see in the blue on this figure. Once we reach the peak, we see that the La Niña-like relationship has disappeared. However, we don't really see a relationship at all. Following the peak, we do see these warm or positive relationships that look like El Niños continue to develop. These kind of peak around 2 years following the peak in solar energy. Because we didn't really see a relationship during the peak solar energy, we looked at the rising and falling solar energy. When solar energy was increasing, we saw more La Niña. When solar energy was decreasing, we saw more El Niño events. In general, we are seeing more ENSO events occurring during low solar periods. It's thought that a connection between the Indian Ocean and Pacific Ocean can cause an abrupt ending to El Niño events and give rise to multi-year La Niña events. This has been kind of noted to happen during low solar periods. On this map here, we can see low solar on this side and high solar on this side, and we see right here this connection to the Indian Ocean is very strong during low solar periods, but is essentially absent during high solar periods. So it's possible that this is why we're seeing more ENSO events during low solar periods. Overall, this relationship between the solar cycle and ENSO is heavily debated, and there's not a very clear consensus on exactly what is happening with it. But if we could find a way to understand this relationship better, it may help us to predict ENSO. This will help these places that are affected be better prepared whenever these events do happen.
How's it going, guys? My name is Mason. I'm going to be talking about my EVS capstone research project, which is going to assess the accuracy of NRCS hydric soil ratings and GIS data in predicting wetland occurrence. So for this project, 2 study areas were selected, and both of these study areas were selected based on their soil properties. As you can see from these red polygons, they were in northwest Austin. So the first study area was going to be adjacent to Brushy Creek and is populated by a hydric soil polygon, as you can see here. And the second study area is going to be more south towards Lake Travis, and this was going to be a non-hydric soil in the Valente silty clay loams. Now, to predict wetland occurrence, the National Wetland Inventory was used to show what aquatic features are in an area. As you can see from here, this pink polygon represents a scrub shrub wetland with a freshwater pond. And so we can say that there would probably be a wetland within this area with a hydric soil. In this study area, there's also a scrub shrub wetland polygon shown here with this pink polygon. And this goes against the hypothesis that without hydric soils, there will be no wetland occurrence. Now, based on the field delineations, we can see that there were, in fact, 2 wetland occurrences in the hydric soil, as you can see from the data point 1 and data point 3 photos. The green polygon shows the field mapped emergent wetland, and the purple polygon shows the field mapped shrub wetland that was found, with the hydric soil shown in each photo. Now, in our second study area, we saw that there was, in fact, no wetland occurrence in a non-hydric soil area, which would prove the hypothesis correct that hydric soil data is accurate in predicting wetland occurrence.
Hi, my name is Evan Legnato, and my capstone research project studies the impact of urbanization on heavy metal concentrations in Waldo Creek. That sounds like a mouthful, but let's break it down. So what is urbanization? In simple terms, urbanization is the increase of human concentration in an area, which leads to the increase of commercial and industrial efforts. When an area becomes urban, the amount of impervious coverage increases. As you can see in this photo, Austin has become much more urban in recent years. The image compares the skyline from 2010 to 2017, along with a population growth of 23%. Impervious cover is a human-made surface that is nearly impermeable and water-resistant, for example, concrete or asphalt. My study area, Waldo Creek Watershed, has an impervious coverage of 50%. This is important to my project because a higher impervious cover percentage means there's a lower amount of natural infiltration that can occur, which leads to pollutants and stormwater runoff that end up flowing into our natural streams. So how do urbanization and impervious cover relate to heavy metals? Heavy metals are naturally found in many ecosystems, but in minimal amounts. Processes like cement creation and cars driving on roads cause heavy metals to be deposited. During a rain event, water travels across an impervious surface, picking up any pollutants that are there, including heavy metals, and deposits them to the nearest water source. Now that you know the basics, let's move on to data collection and analysis. I chose to study 5 metals in particular: chromium, copper, arsenic, lead, and zinc. These 5 metals are commonly found naturally, but are also products of urbanization efforts. To collect my data, I chose 4 points along the creek, starting at the most upstream location, Eastwoods Park, and moving to the stream's mouth at Lady Bird Lake. I collected samples during a rain event in order to get the most accurate metal concentrations. Metals travel and settle quickly, so collecting at the peak of rainfall is important. I also collected a base flow sample to compare my results. Each sample was collected using a collection rod and 1-liter polyethylene bottle. To analyze my samples, I first diluted them and then used an instrument called the ICP-MS, Induced Coupling Plasma Mass Spectrometer. This instrument is able to detect the concentrations of metals based on their ionization. After receiving data back, I compiled them into a spreadsheet to compare it to the National Recommended Water Quality Criteria set by the EPA. From what I've collected, I've proven that for all metals, storm flow values were higher than those collected at base flow. Furthermore, arsenic, chromium, copper all had concentrations above those set by the EPA. So back to the implications. How can metals impact human and environmental health? Actually, metals are very harmful in such low concentrations. Heavy metals tend to be very difficult to break down, meaning they follow a process called bioaccumulation. Marine organisms are susceptible to metals deposited in streams by runoff. Additionally, many metals have been proven to be carcinogens when ingested or accumulated in our bodies. I hope you enjoyed learning about my research project and how urbanization is related to heavy metal pollution. Here's a thank you to everyone who assisted me in this project.
My name is Madison McGee. I'm an undergraduate environmental science student at the University of Texas at Austin. My research has been focused on the impact of leaf litter source on an urban creek. Streams rely on leaf litter fall from trees and other plants as a major source of important nutrients like carbon, nitrogen, and phosphorus. These vital resources feed the bottom of the food chain and support the stream's ecosystem overall. However, in excess, these nutrients can cause eutrophication, harmful algal blooms that consume valuable oxygen in the water and pose a threat to local species. During the largest leaf fall event in North America, occurring in the autumn months, deciduous trees release large amounts of leaves into the waterways as well as the streets. In urbanized areas, these leaves are washed down storm drains during rain events and carried into the streams. These leaves, though dead, retain some of their nutrients, and especially in large quantities can disrupt a stream's water quality. Comparing the species composition and mass of leaves that fall directly into streams with leaves that are carried into streams by storm drains can allow us to better determine how to allocate resources in mitigating stream eutrophication. To study the differences between these two inputs, I collected leaf samples on the bank of Waller Creek, a stream in Austin that runs through the University of Texas at Austin campus, as well as samples from near upstream storm drains that outlet into Waller Creek. Leaf fall began in early December of 2022, and during this month a total of 121 samples were collected across all the sites. The most common species collected from the creek's bank included bald cypress and box elder, as well as American elm, Mustang grapevine, and sycamore. A total of 26 unique species were identified, and the samples were associated with high levels of diversity. In contrast, the most common species collected from the storm drain sites was pecan tree leaves, followed by cedar elm, Shumard oak, American elm, and sycamore. Only 16 unique species were identified in these sites, and the samples themselves held lower levels of diversity than the creek samples. This demonstrates that large amounts of species not local to the creek are being washed into it through storm drains and are contributing to the degradation of the creek's water quality and overall environmental health. Trimming back trees that overhang into the road, as well as properly disposing of fallen leaves during autumn, can go a long way in preventing the overburdening of urban streams with excess nutrients.
Hello, my name is Alicia Rostovan. I'm a graduating senior at UT studying environmental science, and I wanted to talk to you about macroinvertebrates, urbanization, and energy in stream systems. So macroinvertebrates are basically just little bugs that live in the creek. They're normally the baby kind of bug before a bug grows wings and can grow or can fly around in the air. So they're nymph versions, if you will, of adult bugs in their life stages. They could be anything from a fly nymph to a dragonfly nymph, but also included is things like worms and snails. So I want to talk to you about macroinvertebrates because they are a part of the energy cycle in streams. So the way energy comes to streams is from leaves in the canopy around a creek. They fall into the stream, and then the bugs munch on them and break them into small parts before they can be decomposed and used by other function. So the presence of bugs kind of defines the first portion of energy in streams. But something that affects what bugs can live in a stream is urbanization. And that's because it can change the chemical composition of the water, how much water is in the creek, and how it flows. And so learning how cities can affect the energy cycle in a stream, and how rehabilitation projects can maybe mitigate these effects is going to be really important in the future for really using creeks for all they're worth. So I looked at different sites before and after a restoration project on an urbanized stream, as well as a stream that was in a rural area, not near a city. And I put these little bags of leaves out to calculate the diversity and abundance of different kinds of bugs in the creek, but also to calculate how fast leaves are decomposing. So I can kind of look at in each of the scenarios, like before restoration project, after restoration project, and in a rural creek, how bugs and decomposition are related. And my main findings were that most bugs, both quantity-wise and by type, lived in the rural creek, and the least amount lived in the unrestored urban creek. So it's aligned with what you would think intuitively. Bugs like more natural areas. But I found that decomposition was not necessarily correlated to things having more diverse bugs. And this was most likely due to the fast rate of water movement in urbanized streams. There's so much impervious cover around that the streams themselves are more prone to floods, just because water doesn't get to go into the layers of dirt. They instead all wash straight into the basin because they sit on top of the concrete layer instead of being able to run through layers of dirt and rock. There's clearly a lot of knowledge gaps in like what I was studying, just because it's a very complex system. So doing more research to figure out how all these things are related is going to be imperative with figuring out again how to utilize these urbanized streams. But thank you for listening to my talk, and have a wonderful day.
Hello, my name is Becca, and today I'm going to be talking about Central Texas trees, specifically their roots and how they can affect their surrounding rocks and even the carbon cycle. Just like how you and I breathe, tree roots breathe too. This process is known as respiration, and it's a process that consumes oxygen and produces carbon dioxide. When most people think about trees, they think about photosynthesis, which is sort of the opposite reaction of respiration. And this does happen in plants, but today we're going to be talking more about respiration. And plant roots might respire in order to obtain nutrients, in order to grow, or in order to obtain water. Root-respired carbon dioxide increases weathering of rock and leads to short-term carbon sequestration. So here we have kind of a generalized model of a tree rooting into bedrock. And this tree is going to respire and produce carbon dioxide in the form of a gas. However, when rain occurs and infiltrates down into the subsurface, the gaseous carbon dioxide becomes dissolved in the water and is converted into its aqueous form. Now, this aqueous carbon dioxide dissolved in the water lowers the pH of the water, causing it to be more acidic. This acidic water then dissolves away at the rock, eroding it. And what's important about this process is that it consumes carbon dioxide. It sequesters carbon. What is carbon sequestration? It's the process of taking up carbon dioxide out of the atmosphere and storing it in the earth. It occurs naturally, but also humans might sometimes want to speed up this process in order to store more carbon. So for my study, I looked at Central Texas trees and the way that they root into limestone bedrock. So the trees can root directly into the bedrock, often more than 2 meters at depth. So I took 3 measurements. I took surface fluxes of carbon dioxide, which is just directly from the soils. I took gas concentration measurements of carbon dioxide and oxygen, and I also took water content measurements over time. The first main finding is that there's this constant upward movement of carbon dioxide from about 2 meters deep to the surface. So the carbon dioxide that is respired by the plant roots is moving upwards back into the atmosphere. Pretty much generally, this is happening throughout all seasons. However, the second finding is that carbon dioxide concentrations are actually regulated by water content. So the white line here is dry years and the black line is wet years. The top graph is showing carbon dioxide concentrations, and the bottom one is showing water content. So you can see that they are somewhat related to each other. Especially if you kind of dial in here on these red boxed areas, you can see that when water content is high, some gaseous carbon dioxide becomes dissolved in water, and that's portrayed here as the overlapping of these lines at depth. So I hypothesize, based on these results, that during the wet seasons, carbon dioxide is transported downwards and leading to increased mineral weathering. In the future, I want to quantify where carbonate materials are going over time by measuring the changes in their composition. But if we do learn that carbon dioxide is being transported downward and dissolving rocks, we will know that carbon sequestration is happening in the short term. And this is a really exciting result because we're learning about new ways that trees can help aid the process of carbon sequestration. So thank you so much for watching, and I hope you learned something new about our awesome Central Texas trees.
Hello, my name is Summer, and I'm working with Sean Griffin at the Lady Bird Johnson Wildflower Center to study how invasive grasses alter soil properties and water content. So here I listed many reasons why I think these questions are important. Just in general, soils can provide many ecosystem services, and the water in that soil is a big part of that. Invasive plants can choke out native flora and drastically alter the ecosystem that they invade. So this is a little sketch of some of the major fluxes that will be relevant for my project. We have precipitation and infiltration, evaporation, and evapotranspiration from plants photosynthesizing, and lastly, ground heating from solar radiation. So these are the cover types that I will be studying over the course of my project. Although data collection just started, we expect to wrap up around end of June. So the cover types I have are 3 native grasses, Texas wintergrass, side oats, and silver bluestem, as well as 2 invasive grasses, King Ranch bluestem and Johnson grass. So we have 10 to 11 plots of each cover type, about 1 by 1 meter plots. So this project is really focused on a small scale, single plant and soil interaction. These are some data types that we will be collecting and have started to collect. One of those being just characterizing the study area. So that includes lots of things like plant cover, just to understand the habits of these different types of grasses, as well as to learn more about the area that we're studying. The really nitty-gritty of the project is in the soil moisture, soil temperature, soil cores, and light to soil, and these will all hope to get at the question of how a single plant can change the soil. And lastly, we're pairing these with climate data to get a bigger picture of the overall area and to be able to compare each plot at a finer scale. And lastly, we have some implications for this project. So this provides us a greater understanding of how invasive grasses may be altering local ecosystems. And in addition to that, we also have hopefully some insight into how soils and cover type can be associated. And that concludes my project. Thank you so much.
Hi, my name is Dazu, and I'm a senior major in environmental science at the University of Texas at Austin. Today, I will talk about my project that's predicting hurricane activity. First, let's talk about some background information. Hurricanes, also known as tropical cyclones or typhoons, are intense large-scale storm systems that form over warm ocean waters. They are characterized by strong winds, heavy rainfall, and sometimes storm surges, which can lead to coastal flooding. Hurricanes can cause widespread destruction to infrastructure such as homes, businesses, and transportation systems, leading to significant economic losses. They can also result in the loss of human lives, displacement of populations, and long-lasting environmental damage. Here's an example at the Hurricane Ida in New York and Harvey in Houston. So why are we predicting hurricanes? Accurate hurricane prediction allows for better preparation and planning by governments, emergency management agencies, and communities, helping to minimize the impact of these devastating storms. Early warning systems enable timely evacuations, which can save lives and reduce injuries. Predicting hurricanes helps in the allocation of resources. such as emergency supplies, personnel, and equipment, to areas that are most at risk. For my project, my data was from National Hurricane Center and Central Pacific Hurricane Center. And the parameters I chose were sea surface temperature, ocean heat content, vertical wind shear, relative humidity, Saharan air layer, Madden-Julian oscillation, and hurricane occurrences. For model selection, I chose random forest as my prediction model. Random forest is an example learning method that constructs multiple decision trees during training and combines their outputs to make predictions. For example, consider the fruit basket as data shown here. Now, a number of samples are taken from the fruit basket, and an individual decision tree is constructed for each sample. Each decision tree will generate an output here. The final output is considered based on majority voting. In the figure, you can see that the majority decision tree gives output as an apple when compared to banana. So the final output is taken as an apple. Random forest is highly flexible, can handle complex datasets, and is less prone to overfitting compared to the single decision trees due to the average process. For my model training, I use the following steps. First, split the dataset into training and testing sets using a 3:7 ratio. Then standardize the input features to ensure that they have the same scale, which can improve the model's performance. After pre-processing, create a random forest classifier instance and train it on the scaled training data. The model learns to predict hurricane occurrences by constructing multiple decision trees, each using a random subset of features and training instances. Here are some codes that I use to train my model. After lots of training and tuning my model, here's the result. We can see the predicted hurricane activities are the same as the actual hurricane activities. For future work, I will try other machine learning algorithms, such as logistic regression, support vector machines, or neural networks. Also, I need to incorporate additional features, as climate change continues to influence the Earth's atmosphere and ocean dynamics. The underlying patterns and relationships between different factors affecting hurricane formation may change. Updating the parameters ensures that the model remains accurate and relevant under evolving conditions. Thanks for watching.
All right, everybody. Welcome back to our third and final part of our Environmental Research Film Festival. Last but not least, we have student films showcasing geographical research. Talking Trash, a deep dive into international waste and recycling trade with me, Marley O'Brien. All around us, trash is piling up. From its manufacturing to its final incineration, plastic waste is posing as an international pollutant and problem for both humans and the environment. When looking at the facts, about 9% of worldwide plastics end up in a recycling plant. Even then, only around 2% of those plastics will be truly recycled into new reusable products. The other 7% of plastic waste will get incinerated for energy, but most of the time, it just ends up in landfills or in the ocean or traded with impoverished countries. Before 2017, China was the main importer of plastic waste, taking in almost 45% of total exports. Since the plastic import ban in China, this waste was redirected towards much smaller countries, including Turkey, Indonesia, the Philippines, and India, whose imports all increased by over 77%. These countries would buy our mixed plastic waste, pay workers, typically impoverished women, to sort the waste, then sell the usable materials. The increase in imports is simply too much for these smaller countries to handle. This results in a great majority of our plastic waste being burned, which releases very toxic chemicals into the atmosphere. These chemicals are ingested by humans, animals, and can find their way back down to the ground via rainfall. Every year, about 8 million tons of plastic waste escapes into the oceans. Plastic often contains additives, making them stronger and more durable, but these additives can extend the life of products by nearly 400 years in the landfills. A study done in 2019 at a farm near a recycling plant in Indonesia found that 100% of the chicken eggs contain PFOs and PCBs and other forever chemicals found in plastics. The environmental effect of plastic is overwhelming, from fish ingesting microplastics particles in the oceans to PFOs being found in groundwater aquifers. There are very few places on Earth that plastic has not touched. The lobbyists for oil companies are actively pushing for looser regulations of the international waste trade cycle, because as soon as the US has to stop exporting the waste we produce, we will have to deal with it here. Organizations like the Basel Convention are working with countries to create and accept standards for international waste and recycling trade. Their goal is to significantly reduce the amount of plastic that ends up in the ocean and create guidelines for better handling and trading of hazardous waste like plastic. These standards are agreed upon and openly documented. There are 187 UN member nations that are a part of Basel Convention, only 6 UN members that are not: Timor, Fiji, San Marino, South Sudan, and the United States. What should we do? I don't really know. It would take a big change in society and in the plastic industry to make something happen. Ideally, new laws and regulations will be put in place to ban certain types of hazardous chemicals being put into plastics and encourage a more circular system. But for now, reducing our consumption of plastic materials is the best start.
Exposure to some level of air pollution is virtually ubiquitous. On average, air quality here in the United States is not poor enough to be noticeable. However, not everyone is so lucky, and the consequences of air pollution on human health and the environment are measurable and can be observed all around the world. Short-term health effects include eye, nose, and throat irritation, headaches, and nausea, while long-term health effects are numerous: cardiovascular and respiratory disease, cancer, infertility, birth defects, and cognitive impairment, not to mention the environmental consequences, which range from the contamination of water and soil to the rise in global average temperatures, since many air pollutants are also greenhouse gases. The presence of air pollution is indicative of extensive urbanization and industry. Traffic-related emissions, electricity generation, and construction are the most common sources of everyday chronic exposure to air pollution. The UT campus is flanked by 2 of the most congested corridors in Austin, Guadalupe Street and I-35. Our Eckhart power plant, while internationally awarded for its efficiency, still produces 240,000 tons of CO2 every year. West Campus and the UT campus itself are characterized by constant construction activity. In your commute to work or school, just how much air pollution are you exposed to? Do you walk past construction sites or cross high-traffic roads? Do you drive or take public transportation? Would you alter your commute if you knew that you were being exposed to high levels of air pollution? Does it matter to you at all? With the goal of evaluating our community's understanding of local air quality, this study will compare the perceived and actual measured air quality of the UT Austin campus community. I'd like to know if members of the community know where air pollution levels and risk of exposure are the highest, if there is variation in air quality at all, and if they would modify their behaviors to avoid exposure. This could provide a basis for educational campaigns, quantify support for air pollution interventions and solutions, and bring attention to air pollution as an issue in need of action in our community and across the country. According to the World Health Organization, at least 92% of the world's population lives in areas with dangerously high levels of air pollution. Local scale studies are key to raising awareness, identifying solutions that meet the needs of communities, and finally cleaning up our air.
Hi, my name is Carla, and today I'll be talking about my senior capstone project on the geographic history and environmental justice implications of carbon capture and storage development in eastern Louisiana. So first, what is carbon capture and storage, which I'll refer to as CCS, and why is this technology garnering so much attention as a climate solution? So as global temperatures rise and the climate crisis becomes a more imminent threat, governments and corporations are beginning to set net zero goals through which they commit to reaching a target reduction in greenhouse gas emissions that cause global warming. One popular solution to get these emissions down is CCS, a process in which a stream of carbon dioxide from industrial sources is separated, treated, and then transported to a long-term storage location. However, I argue that technologies such as CCS appear promising in reducing emissions, but in practice can be dangerously dismissive of the social costs of their implementation and of the historical inequities that they can perpetuate. To illustrate this point, in my research, I created a series of maps focusing on a stretch of land in Louisiana known by many aliases like Cancer Alley by environmental justice activists because of the heightened cancer rates linked directly to industrial pollution, or the industrial corridor by the oil and gas industry. This region is being targeted as an emerging CCS hub where development would sprawl and environmental activists worry would perpetuate the life of the fossil fuel industry by allowing continued extraction of resources. As you can see in this map, the bright yellow strip in eastern Louisiana contains the densest concentration of emissions from individual facilities. But what's one of the reasons this industry has thrived in this region and accumulated so many emissions? To answer this, we can look back on the state's plantation economy, which thrived in this very region throughout the 19th century. To better understand the spatial relationship between current emissions and the plantation economy, I mapped historical sugarcane plantations throughout the region and even found various instances in which land was sold directly from plantation owners to the new coming industry. But why does this matter today? So, to this day, lower income African American people continue to live in these polluted areas at a disproportionate rate, which can be traced back not just to the plantation economy, but to the history of Jim Crow segregation, redlining, and exclusionary zoning. In this next map, I plotted regions which the Environmental Protection Agency has designated as environmental justice communities for the disproportionately large pollution and health risks associated with living there, overlaid by the industry and plantations we saw before. In this last map, we see emerging CCS development in this very same area, and we do not know if the voices of community members were heard in the process of siting. It is clear then that the industrial corridor through which individual CCS facilities and their corresponding pipelines are being proposed has endured a centuries-long history of oppression and now environmental injustice, and that the current narrow focus on climate and emissions is reminiscent of traditional environmentalism, which was exclusionary in nature. So we can be critical of climate solutions like CCS, not as malicious, but as being currently dismissive of the racist history that has led them to be where they are, so that we can expand the conversation of what true climate justice can look like.
Hello, my name is Maisie. I'm a senior environmental science major at the University of Texas at Austin and will be graduating with my bachelor's degree in environmental science with a focus in geography in May. Today, I'll be presenting my senior capstone project, Fire Dynamics in a Savanna Ecosystem. First, we'll go over objectives, then methodology, region comparison, variable relationships, and then a conclusion. This study examined variations in fire patterns across the entire country of Botswana from 2001 to 2021. Specifically, the Okavango Delta and Central Kalahari Game Reserve regions were compared to each other and to trends across the country. In addition, this study analyzed the relationship between day of year in area burned, and amount of precipitation in area burned. I used 3 separate programs to complete this study. The first part of the study was conducted in Google Earth Engine using JavaScript to process rainfall and burned area data obtained from the MODIS Burned Area Monthly Product and NASA's Monthly Global Precipitation Measurement. I then used QGIS to create maps to show the geospatial representation of the data and to highlight geospatial relationships. Finally, I used Excel to create charts symbolizing the data I analyzed and the relationships I found between variables. Results of the data analysis indicate that fire regimes differ across various regions in Botswana. On this page, you can see maps of the entire country of Botswana, showing the number of times each pixel in the country burned over the study period 2001 to 2021 on the left, and median rainfall per pixel over the study period on the right. Here, we will zoom into the Okavango Delta region in the northwest corner of Botswana. The pixels in this region were burned more often than the pixels in any other area of the country. However, these pixels experienced some of the highest median rainfall in the country. In contrast, the Central Kalahari Game Reserve pixels were burned multiple times during the study period, but experienced very little rainfall. Results of the data analysis indicate that of all the years in the study period, 2011 had the greatest burned area at around 170,000 square kilometers. Interestingly, as you can see on this slide, 2001 is not a precipitation high or low compared to other years in the study period, which can be seen in the time series in the bottom right-hand corner, suggesting that other factors may be at play in the amount of area burned in 2011 compared to other years in the study period. Results of the time series by day of year for precipitation and burned area show a negative correlation between the 100th and 250th day of the year. As precipitation declines between the 100th and 250th day of the year, the number of pixels burned on each day during that period increases, with maximum burned pixels occurring on the 274th day of the year. However, correlation here does not definitively indicate causation. In conclusion, the results of this study indicate that two very different fire regimes exist in the two regions studied closely. We also found that 2011 had the most burned area, but our time series results suggest other factors may be at play in determining fire regimes when Botswana is experiencing the most fire. The results of the time series by day of year indicate a negative correlation between precipitation and area burned. Future directions of research include investigating why 2011 had the most burned area, what other factors are in play in determining fire regimes, and what other factors might be creating the negative correlation between precipitation and area burned in the time series by day of year.
This capstone research focuses on good and bad fires in the Mexico states of Michoacán and Jalisco during 6 different dry seasons throughout the 21st century. Michoacán and Jalisco are of high focus when looking at fire, agriculture, and preservation, as they are the only two states from Mexico allowed to export avocados to the United States. Avocados are a commodity with a growing demand and a driver for land cover change. A cheap tool to facilitate this change is fire. Fire has been used for centuries among farmers to clear and maintain landscapes for agriculture. There are 4 types of vegetation fires: natural, accidental, maintenance, and deforestation fires. For this research, we will only focus on maintenance and deforestation fires, as natural fires and accidental are hard to distinguish from satellite data. Good fires could be categorized as maintenance fires using croplands to help rejuvenate fields, and fires in rangelands that do not occur between dry seasons. Bad fires characteristics are maintenance fires in rangelands that occur between dry seasons in the same area, as it limits the natural succession of an ecosystem. Deforestation fires because they create a risk to biodiversity, and fires in a forested area during a dry season because of the increased risk of a high-intensity fire outbreak. Some of the data collected was done using Google Earth Engine code editor to gather a burned area dataset for Modus Fire CCI and a Sentinel-2 Esri land use land cover change. The first analysis done was a reclassification of the land cover classes from the Modus dataset to align with the land cover classes of the Sentinel-2 Esri land use land cover change.
Now, looking through each dry season, forest fires are common across the entire states of both Michoacán and Jalisco, while cropland fires are closer in proximity to the main road that you see going through the two states.
The last 2 analyses were done using ArcGIS Pro Change Detection Wizard. Change Detection is an image analysis tool that allowed us to compare multiple rasters at the same extent at different points in time. Deforestation is a global issue and its problems do not differ in Mexico. Deforestation is used as a means to convert forest to croplands and rangelands in the beginning stages of commercial use. This is why on the table, majority of the area that was changed was to croplands and rangelands in both states. What counts as bad fire accounts for more than 50% of burned area in each dry season. In a region where fire-dependent agriculture production systems are ubiquitous, they lack proper forest management. Forest conservation and growing agricultural frontiers are at the head of these 2 states because of economic growth over preservation.
Hi, my name is Catherine Christian, and I am a senior EVS major with a focus in geography and marine biology. My EVS capstone project is called Analyzing Trends in New England Aquarium Loggerhead Fasting Behavior, and my supervisor was Dr. Christopher Biggs. So an introduction to female loggerhead sea turtles is that they undergo fasting periods during migration for a year, and female sea turtles in captivity have displayed similar fasting behaviors to those in the wild. I conducted my research at the New England Aquarium on the 2 resident loggerhead sea turtles in the tank, Carolina and Retread. So salinity is the primary variable in the giant ocean tank that gets changed and manipulated. They do this to treat the algae that grows on the artificial reef in the tank. And my study was a comparative analysis of the differences in fasting and behavior between Carolina and Retread and how that correlates to the relationship with salinity concentration in the tank. So as you can see, those are Carolina and Retread on the left. The giant ocean tank is in the middle, about 23 feet deep, 40 feet wide. It's a 200,000 gallon tank. And this is a picture of them monitoring the turtle health because animal welfare does come before all else at the New England Aquarium. And that's also why they let me conduct this research project. So this is more information on the giant ocean tank. It has around 1,000 animals. And some more information on the 2 turtles is that they're both cold stunned and rescued off the coast of Massachusetts. So they do have reduced foraging capabilities, but they still do undergo that annual fasting. So the methods of this study was that over the course of 5 years during the turtle fasting period, data was analyzed in comparison with the salinity levels in the tank and how much Carolina and Retread ate, as well as their weight, their age, food per gram that they ate year, month, and moon cycle. So these are the results. As you can see, 2015 and 2016 had very similar salinities and also lower levels of total loggerhead consumption. And when salinities were on the lower level, there were generally higher food consumption periods. So these are some graphics to explain that further. All of this was input into R, and there is specific coding, a lot of some more complex science stuff put into there. This is another correlation model, basically showing that salinity and food consumption did have high levels of correlation. Some more of that. And basically a discussion, and overall, there wasn't too much of a correlation between the salinity and the fasting. Food consumption was heightened during the years when salinity was low, but overall this information could be useful for aquarium management regarding loggerhead food consumption.
How spectacular. I had so much fun learning about all the different research projects my peers have been a part of. I hope everyone enjoyed the show and learned something new. Huge shout out to all the students who conducted this research and made these films. I'm beyond impressed and so proud to be part of such an awesome group. And a special thanks to our professor, Dr. Haley Gillespie, for putting on the show and leading the charge. I hope everyone has a great rest of your night. Bye.